Okay, hello and welcome to the ZOOMinar today. We have 5 speakers for today. So today's episode is going to be slightly different in format. Through today's presentations, we will try to cover most, if not all, aspects of thioflavin T or ThT-based fluorescence experiments that are very commonly used to study amyloid aggregation. ThT experiment is very simple, easy to perform, and often is the first experiment carried out to study the aggregation kinetics for proteins. But sometimes there are issues with data reproducibility, and even if the data is reproducible, reliability of the data can be a concern. These issues are going to be discussed by the speakers with some examples today, and I'm sure that solutions to the issues will also be provided. We have 5 speakers, and all the presentations and Q&A discussion will be completed within 90 minutes or so. Today's speakers are Dr. Sara Linse from Lund University. She'll be giving a presentation for 20 minutes, and followed by Dr. Steve Bourgault from University of Quebec in Montreal for 20 minutes, and Dr. Sandrine Ongeri from the University of Paris, Saclay for 5 minutes, Dr. Lucie Khemtemourian from CNRS and University of Bourgault for 10 minutes, and finally, Sam McCalpin from Michigan for 10 minutes. Please post your questions and comments in the Q&A folder. We'll discuss them at the end, and you can also join the panel at the end. So with that, I would like to invite Sara, and can you please start your presentation?
Okay, thank you very much, Rams, for this kind invitation. This will be interesting to see where we go with this row of presentations. So I will probably most in my presentation emphasize the fact that thioflavin T is an indirect method, just so that we don't forget that. I mean, when you follow amyloid formation, you can use direct methods. For example, NMR spectroscopy, as you see here, you can monitor the disappearance of monomers, which is kind of a mirror image of the formation of fibrils. And then you actually measure directly on the monomer. You can use intrinsic fluorescence. So amyloid actually give rise to an intrinsic fluorescence signal when they form. That's also direct method, because then you monitor directly the fibrils that form. You can also use circular dichroism spectroscopy because you get a very different spectrum for monomer and fibril. And all these 3 are examples of direct methods that you do in situ. So you can do them in the sample as the reaction progresses. You can also do direct ex situ, which means you have to withdraw aliquots of your reaction. For FTIR, you may need to dry them down. Cryo-TEM, you need to freeze them. AFM, you need to adsorb them on a surface. You can do filter trap assays where you just collect them on a filter with a mesh size that's big enough that monomers and small species go through, but your fibrils are trapped, so you can afterwards quantify them using, for example, radioactivity or fluorescence if you have a fluorophore attached, or other methods. But indirect methods are sometimes preferred. The thioflavin T fluorescence, or you can use other dyes, for example, PFDAA. These methods are often preferred because they are so easy to parallelize. So you can run them in plate readers, in plates, and do 96 or 384. You can do multiple, and you can have multiple plate readers. You can really get a very large chunk of data in one go. Whereas most of the direct methods, as they are developed now, are for few samples. Although, of course, you have sample changes on NMR spectrometer, so you could have a carousel with NMR tubes that you regularly put into the magnet and record. So there are ways to make the others at least semi-high throughput, not very high, but semi. And these are indirect methods you can do in situ. So the fluorescence with dyes you can do in the reaction as it progresses, and it is based on the fact that the dyes have a very different spectrum. Often it's a much higher quantum yield when they're bound to a fibril, which is of course linked to the lifetime. Quantum yield is defined as a ratio of the observed and radiative lifetimes. Ex situ examples you can do also indirect, so trap and seed. You can trap fibrils and then use them to seed new monomers. You can do membrane leakage assays. That's often used to maybe not follow the fibrils, but intermediates in the reaction. Diffusional sizing can also be a very good way of sort of indirectly monitor what's going on. So you measure the average diffusion rate in a sample, or the distribution depending on your setup. But we'll talk here about the thioflavin T fluorescence and the fact that it's an indirect method. But many of the considerations for you to obtain reproducible data are actually shared with the direct methods. So many of them are the same. I mean, the requirement for ultra-pure protein or peptide, we normally study samples that are like 100 microliters and at least a micromolar, which means you have around 10 to the 14 molecules. So that tells you that if the sample is pure, you will get the same kinetics each time you set up the experiment under otherwise identical conditions in terms of pH, salt, agitation, sample container, reading speed, as we will hear later about. So that is a fact that we cannot jump around. So I mean, this is really the key to get reproducible data. Other tricks, of course, is to avoid synthetic peptides. You need excellent pipetting skills so that you don't have to blame reproducibility on yourself, or you get yourself a reliable robot. You need control over all surfaces, including air-water interfaces, and the way the samples are handled during the measurements. But there are some extra that you need to optimize for indirect methods. That means dye concentration, for example, which is the main thing you need to optimize. So maybe also the excitation light, of course. And then don't forget, for indirect methods, you always need to validate, because they are indirect. So you need to validate with something that's direct so that you know what you follow, because of course thioflavin T and any dye, they are relatively fibro specific, but there is no guarantee that they don't bind to something else in your sample. So if we start with the point about recombinant peptides, this is just a mathematical example. So if you do chemical synthesis and the coupling fidelity is 99%, you can calculate by taking 0.99 raised to 42, for the example of A beta 42, that you will have 0.65%. So the yield is 65%. So the fraction of correct peptide is 0.65, and the fraction incorrect is 0.35. So you have 35% crap after this synthesis. And all this crap is 41 different A beta 41. So you can have miscouplings, you can have 1,700 different A beta 40s in this. So there are lots of peptides that are very similar in chemical properties. So they may actually be difficult to purify away from the one you want. You also need to have ultra pure protein or peptide. And what does that mean? So I mean, the first easiest check you can always do, of course, is to check there are no other bands on SGS page. So this is silver stained gels of A beta 40. This is our recombinant A beta 40, side by side of 2 lanes with a synthetic, which looks fine because you don't see racemates, you don't see miscouplings, it is just one amino acid. You don't have that resolution on SDS page. You don't see if there are D amino acids, which are racemates. And this is A beta 42 recombinant synthetic, and this is often called the SDS artifact. So I mean, this is the same for any protein you do. You have to check that there is nothing else to be found on your gel. And if you work with tau, for example, it's super important that there is not a shadow of an extra band underneath, because that massively perturbs the aggregation kinetics. So it needs to be one form, no other proteins. Another, of course, consideration is that if you run an NMR spectrum of your purified peptide, you should only see the peptide. You shouldn't see a lot of small molecules there. If you run a UV spectrum, you shouldn't see DNA. I mean, so you don't want to see non-peptide stuff in your sample. You don't want to see any adducts by mass spec, that you cannot see here. If part of your peptide is oxidized or has for example, if you use old or bad quality urea, you will get carbamylation of your lysines that's easily detected by mass spec. So I would always advise you to run a mass spec of your purified protein or peptide, because mass spec never lies. So it's like, I guess you have, like me, a hate-love relationship with mass spec, because you can believe you have a perfect peptide and mass spec tells you you have not. So your happy day is turned into a sad day. In our lab, we also check each batch, for example, of A-beta, we check each batch that we get the correct scaling exponents. We actually set up an aggregation kinetics over a range of concentrations, and then we compare with what it should be. So it should be like minus 1.3, the scaling exponent, if you plot log half-time versus log monomer concentration. Or you can plot it, of course, non-log, but it's still the same. You fit the power function. So if a batch appears like this, we discard it. So we always do this check before the batches go into the freezer and can be used in any kind of experiment. So in this case, you can, of course, continue to purify, because often just one more round of purification may turn it into a pure batch. And I have often gotten batches from other investigators. They complain and say, we follow your protocol, but we don't get the same kinetics. And then I ask them, please send me the batch. And they do. And I see often lots of other peaks by NMR. I purify it, and after purification, I get the correct scaling exponent. So don't give up, just add purification steps. That will always help. And you can never get around an impure batch by some other tricks. So I think this is the absolute key to any method, direct or indirect. So a hint when you purify them, we often, whatever is the protein, we often do ion exchange first. Sometimes we do HIC, but we always end with size exclusion, and often, like here, a big size exclusion and then small ones. Or so you just do multiple rounds, and the hint is to always sacrifice the flanks, as we call them. Only collect the center from like half to half. Only collect that. It hurts to throw away, but here you have maybe other proteins. You may have breakdown products. So yeah, even if it hurts, you get a better product in the end. And if you do 3 rounds of size exclusion, you may lose as much as 80%, but it really hurts. But the end product is super good and gives you very nice kinetics, always the same. So you just have to learn to live with the pain of losing peptide. And of course, you need excellent pipetting skills. And if you don't have, buy a robot. So in my lab, we actually have a person in my lab, Tom Lading, built this robot for us. So here's the plate. And it has some tubings, and you put your buffer and peptide in different tubes, and you just tell the computer what will be my dilution series, if it should be linear or logarithmic, and how many wells of each. So how many replicas of each solution. And then the robot, of course, just like every column you use needs to be cleaned and cleaned and cleaned. And we use exactly the same. We use formic acid, 50%. We use half a molar sodium hydroxide. We use twist base and a lot of water in between to clean all the tubing in the robot, just like we clean all the chromatography systems and columns in between runs. And you also, of course, for every sample, and every method, direct or indirect, you need control over your surfaces. And one way to carefully degas all your buffers, so you minimize the air-water interface in the buffer. We typically use half area wells. I would like to have bomb wells so that you have no surface area at all, because you always have some air-water interface. We typically use pegylated plates because they have less binding of the peptide. So if you have plain polystyrene, you can have both catalysis and retardation of your kinetics, and it will depend on the concentration of the peptide whether you get retardation or catalysis. So and if you take care of all these things, whatever is the method, you will have nice reproducibility for samples of 10 to the 14 molecules or more. And these are triplicates, example of triplicates at 6 different concentrations. So extra for the indirect methods like thioflavin T is really the optimization of the dye concentration. Of course, you have to optimize the wavelengths, but that's like in all spectroscopy, the excitation and emission. But you want a linear response. So this is example for A-beta 40 from one of our publications, where we did kinetics from 3 to 70 micromolar A-beta, and then you have to check that the plateau value at the end follows a straight line. I mean, there is some deviation, but roughly this is a straight line. There is no systematic trend here. So this you want to see to be sure that you can actually use whatever is the thioflavin T concentration you have decided. And a way to decide is actually to take a sample of fibrils. So first make fibrils of your protein without thioflavin T. So don't follow it with thioflavin T, follow it with something else. for example, CD spectroscopy or NMR, or take an aliquot that you don't follow at all, and then afterwards take out a little bit and add thioflavin T just to validate you have fibrils. Okay, then you have your fibrils, and you titrate in thioflavin T and measure the fluorescence intensity. So you can record the whole spectrum or just the intensity at the wavelength of interest. And in the beginning, the more you add, it will just increase. So here you are suboptimal. So that is the range where the thioflavin T concentration is too low to bind to all the sites in the fibrils. You have to reach a plateau. And then if you over-titrate, I mean, you'll be happy for a while. And then when you over-titrate, you end up in a regime where you actually quench the fluorescence due to the inner filter effects. You have too much dye, and then part of the fluorescence is actually reabsorbed by the dye molecules. So you lose fluorescence intensity. There can be other factors also leading to quenching of the fluorescence at too high concentration. And I think this is true for all fluorophores and all fluorescence assays, that you really can't go too high with the fluorophores. So you end up with some kind of linear regime where if you modulate the thioflavin T concentration in this way, you get the same response. So then you know you are in a good regime. And then you can, in principle, repeat the titration at a range of different fibril concentrations that represent the range that you are going to use in your assay. And then you may find actually a smaller linear regime. So you may have to lower the regime. And then often, what I often do, then I choose a concentration in the middle of the regime. So in the case of A-beta-42, we found this regime over the concentrations we were interested in to be between 6 and 20 micromolar. So we often end up at like 10, 12 something, somewhere in the middle of the regime. We feel happy. You may also want to check that the curve shape is consistent over the entire curve. So if you have too low thioflavin T, you typically get a nice onset. And this is good if the onset is all you need. For example, if you're going to take your fibrils to some post method like cryo, and you don't want to have too much dye around, maybe this is good enough, because then you know they will be formed here and you can pick them. But here we can see that the shape is not correct, because we end up in a situation where we sort of run out of thioflavin T. And then we get a funny shape at the end of the time course. So if you then just add a little bit more thioflavin T into the linear range, if you are in the linear range, this is what you should see. That the end also looks okay. And then, of course, if you have too high thioflavin T, it still looks okay, but it flattens. It flattens at a lower value than it would otherwise do. So then finally, I will just, and why did we jump there? Okay, we should be here. I will just underscore the necessity to validate your method, because thioflavin T is just an indirect reporter of amyloid formation. So this is an example of data from one of our publications. I didn't put the reference here. It is from 2015 in Chemical Science. The two first authors are Risto Chukalevsky and Xiaoting Yang. And in this case, it was mixtures. This was actually equimolar mixtures of A-beta-42 and A-beta-40. And the green here, if you start to look at the green, that is the thioflavin T fluorescence curve that we get. So first they got one transition, then a plateau, and the second transition. And here we could interpret, if we want, from the thioflavin T that this is A-beta-42 aggregating first and then A-beta-40, because we have much higher quantum yield when thioflavin T binds to A-beta-40 than to A-beta-42 under otherwise identical conditions. But to validate, we then use mass spectrometry, or they use mass spectrometry. So, okay, we go here. So they picked samples at the arrow time points, so time 0. And in this experiment, they actually had A-beta-42 labeled with N15. So after taking samples at different time points, spinning away the fibrils, and then checking the monomers left in solution, after digestion, so it's easy to do mass spec, easy to ionize peptides from the N-terminal part, They could see that at time 0, nothing has really happened. They are both at the same concentration. At the end of the lag phase, it's the same. I mean, the monomers, in principle, you have all the monomers left here, although you have formed fibrils, but the fibrils are not above the signal to noise level of the assay. And also the monomers have not gone down below this, what you can detect, what is the signal to noise level of this assay. Same at time 2, you see still roughly the same concentrations. You're still in the early part of the lag phase, but after the transition, A-beta 42 is all of a sudden gone. Now here you can see that some A-beta 42 is starting to be gone, but him and here is really gone. And then at T4, which is further into the plateaus, you can really validate here that the first transition is actually due to consumption of A-beta 42 monomers from the solution. So the supernatant after centrifugation contains only A-beta 40 when you come to here. And then they did all these time points. And then at the end, at the time point 11, both are gone. So the red here is the ratio. You can't really calculate the ratio when there's no signal, but the ratio of the two peptides goes down. So 42 goes down very quickly to 0 here. So I think I would end there. So now we have no more slides. And we go on to the next speaker.
Thank you very much, Sara. The next speaker is Steve Bourgault from the University of Quebec, Montreal. Please post your question for each speaker in the Q&A folder. Don't wait until the end. But we'll discuss all of them at the end. Steve, go ahead. Share your screen.
You see my screen?
Yep.
Perfect. So first of all, thank you very much, Professor Amamorti, for putting this ZOOMinar together this morning, and really thank you to Sara for introducing us for this important thioflavin T-based assay. So what I will be discussing in the next 10 to 15 minutes, maybe 15 minutes, is how sensitive thioflavin T-based assay are, and mainly the one that we're doing in the microplate format. So as you all know, if you're here today, amyloid formation is often described as a polymerization mechanisms that involve 3 phases: the nucleation, the elongation, and the saturation. And a good way to follow this kinetic of amyloid formation is to use thioflavin T, which open binding to this cross beta sheet quaternary motif. There is a sharp increase of this quantum hill. So one of the advantages that this indirect method, like Sara said, is that it can easily be applied to a microplate format. So this is really useful for people who want to generate a lot of data really rapidly, screen effects of different conditions on the amyloid formation. So basically you have your peptide or protein of choice, you put in different wells of microplate, you add the thioflavin T as a specific concentration, and then basically you can have those nice curves, fibril formation. However, those kinetics are highly sensitive to different conditions. So if you look in the literature just for the islet amyloid polypeptide, I mean, you have all kind of lag time. So a lag time that is coming from 1 hour to 3 hours in a similar range of concentration. And if you look also with the A-beta 140, you have everything as a T-lag of 30 hours to a T-lag to 1 hour or 11 hours. So there is really a high heterogeneity, an apparent irreproducibility in THT-based assay. So of course, like Sara said, there are many factors that are affecting this kinetics of amyloid formation. So of course, experimentally, the quality of the peptide, the protein precursor, it's important to start with a nice monomerization monomer. The sterilization, the purity is critical, but of course there are many aspects also that needs to be taken care of: the buffer, the ionic strength, agitation, pH, temperature, the surface, the volume, the ratio between the volume and the surface. So it's important to have that. The air-water interface also, and of course biologically it's even more complex. So in my group, we're working on the islet amyloid polypeptide, and we have established a really robust monomerization and sample preparation protocol Basically, we're doing the synthesis in our lab, so we can purify it 2, 3, 4 times even. We confirm the purity and the identity of the peptide by high resolution mass spectrometry. And then we're going through a long process of monomerization, which involve treatment with HFIP, filtration, sonication, and freeze drying. And at the end, we add up with different aliquots of powder peptide, 100 to 500 microgram that we keep dry at -20, but no longer for 3 to 4 weeks. Every 2 to 4 weeks, we need to repeat the process from the purification and so on. So if we do, if we follow this protocol, so we can get really a reproducible data. So if we do a kinetics in a microplate format, using those conditions that are shown here, so the fixed concentration of IAPP, thioflavin T, buffer, a specific volume per well, in a microplate, of course, the surface is important. Here, it's a non-binding surface microplate. We do an initial mixing, and we have no agitation. So no agitation between each reading. I mean, everyone that get in the lab, and I'm sure that if you follow this specific protocol, you will get something like similar as that with a lag tracer around 4 to 6 hours. But one of my grad students that came to my lab in 2016 observed a really interesting phenomenon. So basically, usually we read the fluorescence every 10 minutes, or some people are reading the fluorescence every 5 minutes, other from every 1 hour. And what it was interesting, what Matthew observed, and it was really fascinating, it's that the interval between each lecture, fluorescence lecture, affect the kinetics of fibril formation. So if you read the fluorescence every 10 minutes, you get the curve in purple. And if you read the fluorescence every 1 minute, you have a sharp increase, an acceleration of the fibril formation. And Matthew then confirmed this with different interval measurement intervals. So you can see if you read the fluorescence every 60 minutes, while if you read the interval every 30 seconds, you have a really different kinetics, different lag time, and you need to consider here that the condition are the same. So we're using this exact same condition here, but the only difference that we're changing is the interval between each lecture. So as you can see here, you can plot those curves through a sigmoidal growth, and you have a nice correlation between the measurement interval and the T50 or the lag time. So of course, thioflavin T is an indirect method. We wanted to confirm that what we observed with thioflavin T was indeed associated with fibril formation. So as you can see here and at the bottom, if you read the fluorescence every 1 minute, after 3 hours, you have already a CD signal associated with the beta sheet, and you have the presence of fibrils. While here, if you're reading every 10 minutes, you need at least 5 hours to 24 hours to get a signal of a beta sheet and also the presence of fibrils. So of course, that means that what we're observing, what was going on in the microplate was actually most likely true. So what is this explanation? So the first thing that we're thinking of is the temperature. So if you read more often, probably, although we're trying to keep the temperature constant at 24 degrees Celsius, or maybe it changed a bit, and the microplate reader is not able to keep the temperature constant. So we try the kinetic at different temperature, and as you can see here, there is not a big change if you change the temperature. You still have the same trend. So 1 minute here in blue and red, and in green and purple, the 10 minutes interval. In the literature also, Matthew went in the literature and he saw a nice paper in 2009 in GBC showing that, laser irradiation through THT can use fibril fragmentation. Then in fact, in turn, it can promote secondary nucleation. So he went back to his essay, so he changed the thioflavin T to another dye. There are many different dye, but here he tried ANS, and the same trend was observed using ANS instead of thioflavin T. And also, he saw that this effect was independent of THT concentration. So if you do the 1 minute interval or 10-minute interval, you have the same trend, whatever the thioflavin T concentration you're using it here. So, but if you perform the same kind of kinetic, but instead of in a microplate format, but in a quartz cuvette or in a cuvette directly in the fluorescence in the spectral fluorometer, you don't have the same difference here. If you measure the fluorescence every 1 or 10 minutes at every 1 to 10 minute interval, you have basically the same kinetic. So it's really related to the microplate format to the reader. So Matthew's done a really clever experiment. In one plate, he was reading only 3 wells. In the other plate, he was reading the entire plate here. But of course, the data here that I'm showing you is the data for the same 3 wells. So as you can see, if you read only 3 wells in one plate, you have a kinetic that is quite slower compared to if you're to a plate that you're reading the entire plate. And this phenomenon is even more pronounced for the A beta 140, as you can see here. If you have a microplate that you're reading only 3 wells every 10 minutes, you will have practically a really weak, or in fact, the nucleation poly will just start a bit later. While if you read the entire plate here, you will have like a nice kinetic of fibril formation with a lag time of around 12 hours. So what's going on? Basically, it's just that the way those microplate readers are set up is that they move the plate between each well. So they move the plate to do the reading. So just the fact that the plate is moving inside the microplate reader is sufficient to significantly alter the kinetics of fibrillization. And basically what's going on is then when the plate move between each well, between each reading, basically you increase agitation and you further primary and secondary nucleation. So again, here in summary, it's just showing you, it's a small example showing you how much amyloid formation is sensitive to agitation and also to the conditions in which you're setting up the assay. And basically, if you look in different papers, the number of wells that was read per plate is usually not something that we think that is important, but it is. And also the measurement interval is a critical factor. So the kinetics of amyloid formation in microplate requires really strict methodology for reproducibility. So of course, I would like to highlight that we published this paper a long time ago in 2017, but feel free to go and see this paper showing how sensitive this assay is. And I would like to definitely thank Matthew, a PhD student in my group, that made this observation. Of course, the fact that the plate is moving in a microplate reader is definitely not his PhD project, but it was a nice side project, and I think it's an important contribution to the field. Thank you very much for your attention.
Excellent, Steve. Thank you very much for the nice presentation. Next speaker is Dr. Sandrine Ongeri from University of Paris-Saclay. Sandrine, can get started.
Yes, thank you, Rams. Do you see my screen?
Yes.
Okay. So I will show you the issues of reproducibility we have according to the sources of amyloid peptides. We have a huge problem with tau, A beta, alpha synuclein, and IAPP according to the sources of the amyloid peptides. It's not a problem of experimentator because only Julia Caffey is doing that. So she's associate professor in my lab, and she's always doing exactly the same conditions, the same buffers. Everything is always the same. and you will see that it's very different according to the batches we have or the suppliers. So here I show you some experiments with tau, and the first problem we had was to reproduce the THT curves described in the literature. In particular, you can see here, for example, that usually the heparin is necessary to induce the aggregation of tau, and usually a small amount of heparin, tau, the ratio of tau heparin is about 4:1 or 2:1, and here in our hands, this was not able to gain and to obtain very reproducible curves. So we had to optimize, and Julia found that a large excess of tau and a very small amount of heparin was good to obtain these blue curves, so very reproducible curves. When we changed the batch, so in these two batches were produced by colleagues from our tubing train network. So in that case, the first batch was produced by colleagues in Switzerland, and here from colleagues of Osnabrück University. And you can see that the lag phase was shorter, but at least the curves are reproducible. But even according to the batches, the lag phase was very, very shorter. In the case of A-beta, for many years, we didn't have any problems. We bought some A-beta from American Peptide or from Bachem, and you can see that we were able to obtain very reproducible curves, even at different temperature, even at different time, or according to the years. So we didn't have any problems. Why? Since 1 year, we have met many problems of reproducibility. You can see here, for example, with different batches from Bachem, that the lag phase is very, very short. And from peptide obtained by recombinant techniques from air peptide, in that case, we had already some aggregated A-beta 142. So we decided to use this batch and to try to treat it, to perform pre-treatment to try to delay the lag phase, because in our case, we are medicinal chemists and we design compounds able to target really monomers or small oligomer species. So in that case, our compounds were not anymore active, of course, because here we have already too large amounts of oligomers or fibrils. So we tested different pre-treatments, no pretreatment with ammoniac or with HFIP, with or without sonication, and we observed that there is no difference between sonication versus no sonication at 5 minutes or 1 hour. The pretreatment that was used before by us with the other batches was not anymore efficient, and only HFIP allowed us to recover a longer lag phase. In the case of alpha-synuclein, we worked using different protocols described, for example, here from the work of Gazit, who was using PBS and at pH 77.6. And in our case, the lag phase was too long. You see that we observed the aggregation of alpha-synuclein only after 8 days, which is very too long. So we developed other techniques. We tried acetates because acidic pH can increase the aggregation. And finally, the best medium was PBS10X. Then we tried to sonicate. And in this condition, we observed a decrease of the lag phase, but no reproducibility. So we Julia developed many other techniques to try to obtain reproducible curves, and she tried sonication, heating. This was better, and finally, by adding Teflon balls, she could obtain quite reproducible curves, and the lag phase was increased. Only 5 hours while without Teflon balls, it was more 5 days. And to finish with the IAPP, so this was published by one of my PhD few years ago. We evaluate the differences between batches. So we had three different suppliers and 2 different batches from one supplier. And you can see here that the THT curves were very different according to the batches. For example, the lag phase was 2 hours for batch A2, while it was 7 hours for C, and we had also a big difference of the plateau. And when we checked the oligomer, the oligomerization, so the early steps of the aggregations by capillary electrophoresis, you can see here that we observe many differences between the 3 suppliers. Here you have the monomer species at T0, and it decreases a little bit. After 10 hours, you have almost no more monomers in the case of batch A, while here you have almost no monomers from the beginning, and you have a large amount of oligomers very quickly. And in this case, you can see that after 20 hours, we still have a lot of monomers. So in fact, the aggregation was completely different. The oligomerization was completely different. So we had a look on the monomers and dimers by MS, by mass spectroscopy. And we could see that according to the batch, we have many different conformers. So the batches are very important, the suppliers are very important, and the conformation of the monomers are very different and very important for the aggregation, for the first oligomerization step, and for the aggregation. So we need really to select carefully, especially when we are medicinal chemists and we don't produce by ourselves the peptides. So we have really to check the batches we receive, to monitor the oligomerization process and its further fibrillization. This was for the 4 amyloid peptides we studied. I think.
Are you done?
Lucie can speak now.
Are you done? Oh, okay. Thank you very much, Sandrine. Next speaker is Dr. Lucie Khemtemourian from CNRS and University of Bourgault. Lucie, get started.
Sorry, I make a mistake. I will share it again. Do you see it or not?
Yeah, I see it. Go to the presentation mode.
Yes. Like this. Okay.
Yep.
It's okay?
Yep.
Okay, so I'm going to talk today about IAPP fibroid formation using THT and the problem we had in our lab recently. So just well, maybe you don't know, but IAPP is a human direct amyloid polypeptide, which is a main component of amyloid fibrils found in pancreatic in the pancreas of type 2 diabetes patient. And in my lab, we use a synthetic IAPP. Sorry, I'm going to put. We are using a synthetic IAPP, which is I made it by myself. My student made the homemade IAPP using the protocol which is described by Dr. Antiche Abedini, or we bought it from Bachem. The preparation is the following. We dissolve it in HFIP and we incubate it for 1 hour. Then I evaporate HFIP by blowing nitrogen gas, and I put it in desiccator for 2 hours. And depending on the experiments, I dissolve either in DMSO 1 millimolar and then in the buffer at 0.2 millimolar, or directly in the buffer at 0.2 millimolar. Then for the THT measurement, I use a 96 black flat bottom well plate, a transparent cover stick. I check 10 seconds, double orbital for 500 RPM. I measure every 10 minutes and I do a flash. I do between 8 to 50 flashes. And then I use a concentration of 5 micromolar of IAPP and thioflavine 10 micromolar. And during the last 10 years, I kind of get the same reproducibility. So with 5 micromolar of IAPP and 10 micromolar of THT, in this buffer, I have a T-alpha of around 3 to 4 hours, which is here, here, and here. But recently, since 1 year, we observed that IAPP fibroid formation was and is still quicker. We have a T-alpha for 1 hour for both peptides, the one we made in my lab and the one we bought in Bachem, using the same condition as previously. So we also want to have a nice lag time because I'm also testing some molecules to check if there is inhibition. So I really want to have a nice lag time. So this is a bit quick for me. So I look at different parameters that I could change to increase this lag time. So I try to incubate in HFIP overnight instead of 2 hours. I change the ratio IAPP-THT from 1 to 10 to 1 to 20. And I try to dissolve it in a buffer or in DMSO. And finally, here we can see the difference between the incubation in HFIP overnight in 2 hours. It's more or less the same T-alpha. We have around 1 hour each time of T-alpha, so it doesn't change so much. If I increase the ratio IAPP-THT, I have again a T-alpha of 1 hour, so this also doesn't change so much. And if I dissolve in buffer or DMSO, I also observe again the T-alpha of around 1 master. So it means that doesn't change. So all the conditions I tried since 1 year always gave me the same T-alpha of approximately 1 hour. So with the RAMS, we decide to test in our both lab the kinetic of IAPP fibroid formation in the presence also of zinc and insulin. So we do the same experiments, either the same experiments in Michigan. For some does the same experiments in Michigan. I'm doing the one in Bourgault. We use different IAPP and different plate reader, of course. And we are looking at the effect of zinc and insulin, and we are comparing our results. So I am showing you now my results, and you will see just after that some has quite similar results. So it's quite positive because we are able to reproduce similar results in both labs, even if IAPP fibrillation is quite fast since a year. So this is a result in the presence of zinc. So if you look a bit more in details here, you can see that zinc increase the T-alpha and decrease the maximum of intensity in the concentration-dependent manner. The T-alpha is going from 1 hour without zinc to 2.3 hours with 5,000 micromolar of zinc. And we also see a plateau reduction. If we do the same experiments with insulin, which is known to inhibit IAPP fibroid formation, we also see here in the first event of the kinetics, that insulin retards IAPP fibroid formation. And interestingly, what we see also a second aggregation between 20 to 30 hours. So we hypothesize that this aggregation comes from insulin, and THT is able to finally bind to insulin aggregates. And we did some microscopy images, and indeed we see some fibrils. Well, not fibrils, but we see really huge aggregate. So we can see that the second, we hypothesize that the second step is due to insulin. And but Sara just gave me an idea to do some mass spec to really confirm this hypothesis. So I think I will do this next. And then what we wanted to check, we want to see if zinc and insulin together had an influence on IAPP fibroid formation. And here you see that the insulin, we keep the concentration of insulin constant at 2.5 micromolar, and we increase the concentration of zinc. And we can see that the T-alpha of IAPP increase at the beginning, so suggesting that insulin does inhibit IAPP fibril formation. But after a while, we have the opposite effect. So we hypothesize that zinc interacts with insulin, and then we have less insulin available to inhibit IAPP fibril formation. So we did the same experiments at 5 micromolar of insulin, and we see again the same behavior. So at the beginning, insulin inhibit IAPP fibril formation, and after a certain concentration, there is no inhibition anymore, probably because zinc interact with insulin. So Sam is going to present the work he has done in Michigan to see how the results are reproducible. So, but the take-home message for today is that since a year we have the IAPP fibril formation is faster than before, so we have at around 1 hour. It's independent of the preparation of IAPP, and now Sam will present the work he has done and to show you that we have the same results between 2 labs. So Sam, it's your turn.
Okay, thank you, Lucie. Next speaker is Sam McCalpin from my lab, Michigan. Go ahead, Sam.
Thank you, Rams. Okay. Right. So as Rams and Lucie have said, so I'm a student in Rams's lab. I've been working here about doing this experiment for about a year or so. So I don't have the sort of pedigree or experience that some of the other members of this panel have. So I want to come here and just give an example that Lucie introduced about how I have worked through some of these issues that we've come up with this THT experiment, and hopefully just give you all some things to think about rather than, and maybe some general suggestions on how we can go about optimizing this experiment. So when I first started in the lab, I was sort of on my own. Everyone from the lab who had done this experiment for us before had left. So I was sort of, working on this myself. Yeah. And I had, yeah.
It should be on the presentation mode. This is presenter's view, I see. Yeah, good, that's good.
Okay.
Sorry.
My apologies.
Yeah, go ahead, please.
Right, so what I was saying, so when I first started working through this experiment on myself, we had several issues. We had these really ugly curves, which don't look sigmoidal. They're really like noisy and variable. And so the previous, like Dr. Linse and some of the previous speakers have spoken to some things that can improve the quality of that, especially the quality of the peptide. But so what I'm going to focus on here today is this issue we had where we would do these experiments in replicate, but the error bars that we would get, the variability across the replicates was huge. And so from my perspective, this was the first issue we should tackle, because when we have error bars like this, so much variability across the replicates, we can't really interpret the data at all, even to the point of saying like how the curves look wrong or not or whatnot. So what I'm going to go through here is how I've worked through this system of troubleshooting the THT fluorescence experiment parameters for it, the system that Lucie introduced, the IAPP, insulin, and zinc, and then compare a little bit to some other systems that I've done this experiment for, where different conditions have given reproducible data, and then maybe offer some general comments and suggestions on some things to keep in mind maybe when we're thinking about the things, the parameters of this experiment and how to go about performing it. So as Lucie introduced, we were studying the system, this IAPP, insulin, and zinc, 3-component system. And so I'm going to talk about 3 parameters in particular that I worked through to see how these affect specifically the reproducibility of the data. So this was the first set. This is the basic how our lab had been doing things before. So just everything dissolved in aqueous solvent, no agitation, 3-minute measurement intervals. And so the issue we had here is that you can see, so these are all replicates. So for 20 different sample conditions, we have 3 replicates. And you can see for several of the conditions, it's not just that there's variance in the lag time and the fluorescence intensity, but in some cases, the shapes of these curves aren't even the same. And so we don't think that these curves, it's hard to trust that this data is trustworthy or useful because these are the same sample prepared just in different wells, and there's so much variability in the shapes of the curves and how they look. So the first thing I tried was, so this was what Lucie had been doing beforehand. So some people like to dissolve their peptides in DMSO or another organic solvent beforehand. And this is especially, we wanted to try this because insulin is sort of hard to solubilize at the pH we were looking at. And so we can see here, there's an effect on the kinetics. The lag times, I think, are a little longer. That second step in the profile has gone away, but there's still some issue with in several of the samples, the shapes of the replicate curves just don't even look the same. And so I wanted to try something else. Maybe we came across Dr. Bourgault's paper about the measurement frequency, which I was really happy to read because that's not something I ever would have thought about before. And it turns out, so increasing or decreasing the measurement frequency, increasing the interval between the measurements seems to have a pretty dramatic improvement on the reproducibility of the shapes of these curves. So there is still some, variability in the lag times and fluorescence intensities, but for the most part, these shapes are all the same. So as Dr. Bourgault was talking about, this parameter does affect the kinetics of the experiment pretty significantly, and in my hands at least, it also seems to improve the reproducibility of the data. And so then the last thing we wanted to try was whether shaking. A lot of people do their experiments with shaking, and that the homogenization of the samples with the shaking conditions might improve the reproducibility. And so the takeaway here, I think, is just that, again, the reproducibility isn't atrocious, especially in the short time frame, but there's still some variability across the replicates that's not ideal, and the lag time is so short that it's not really easy to interpret any differences there. So I came to the conclusion that, in my hands at least, the optimal conditions for at least these 3 parameters, there are a bunch of things that I, of course, didn't try, is that this longer measurement frequency with no organic solvent and no agitation. And we can see that we get, when I average the replicates and put the error bars on, the curves may not look perfect or totally aesthetically pleasing. I think some of the previous speakers have given some good suggestions about how we can improve that. But we can see that the curves are clearly distinguishable from each other. The error bars don't overlap, and we can clearly see the differences between the different conditions. And as Lucie said, we between our 2 labs, we generally see the same thing. So we were able to, it seems, solve this reproducibility issue just by systematically going through changing the parameters and deciding what's best. I do want to point out, though, that So this was a different experiment, totally different system, A beta in this lipid molecule called GM1. And I found doing the same experiment with totally different conditions, I was able to get pretty reproducible data. So here we are shaking, the measurement frequency is about the same, but it's different temperature. They're shaking, it's a higher concentration of peptide and sodium and THT. And for both 40 and 42, we were able to come up with pretty reproducible data across replicates. So, and similarly, this paper from Dan Utsen's group shows that for alpha-synuclein, at least shaking improves the reproducibility of the assay. So the point I want to make there is just that there's not one set of conditions that's going to work for each system. You're going to probably have to optimize for each experiment that you're doing, unfortunately. So I can make, at least in my hands, some specific conclusions about the system I was working with, the IAPP, insulin, and zinc, saying that we probably don't want to use the organic solvent, and that agitation has a pretty significant effect on the variability in the data we collect. I think it's interesting to note that there's a difference between the orbital agitation, which is the shaking generally that these instruments do, versus linear shaking, which is more what's happening when the measurement is occurring. The plate is being moved in linear ways. And it seems like this linear shaking introduces more or has a more detrimental effect on the reproducibility. So just to like summarize some things, maybe some ideas from all of the presentations today. I think obviously there's a lot of optimization here that you're probably going to have to do for each batch of peptide, each set of sample conditions, and you're not going to come up with the same experimental parameters or instrument protocol each time, and that you're probably going to have to put time in to optimize that. I do have this sense sort of that because so many of these factors influence the kinetics of the experiment, I wonder if there's maybe a sort of sweet spot. So when you're choosing your parameters, whether they increase or decrease, accelerate or inhibit the aggregation, maybe if there's this sweet spot in the lag time or the kinetics of the aggregation that will give the most reliable or interpretable and reproducible data. Not to say that you can't have data outside this range that is good, but just that maybe there's this sweet spot that might be something to shoot for. And I want to reiterate something Dr. Bourgault said in his presentation, or hinted at in his presentation, that a lot of these things that we are learning affect the data we collect in this experiment are not often included in the experimental sections of publications. And so I think it's really important that moving forward, we are really deliberate about including all of the considerations, all of the details of the experiments and how they were performed, even the things that we wouldn't think otherwise would have much of an effect. So that's all I got, Rams, if you want.
Thank you very much, Sam. Thanks to all the speakers for taking time out and sharing their data and experience. And we will open up, we're going to open up the session for Q&A. There are 2 questions in the Q&A folder, but please post your questions or raise your hand to join the panel. You can discuss your experience or post comments to the speakers directly. So first question for Sara. Is Sara here? Maybe she's not here. Okay, great. So if you can look at the Q&A folder, I'll read it for you. The first question
Yes,
is
there is Q&A, yes.
Do you naturally present the best way to get clean kinetics for small amounts of ultra pure peptide? Can you comment on ways you would scale up to a large sample, milligram amount, for example, solid state NMR samples? will maintain.
Yeah, I can comment on that because we have made several samples for solid state NMR. So the first one we made was 70 mg of doubly labeled A-beta 42. So it was carbon 13 and 15. So we actually do, so the protocol we use, because it's based on ion exchange in a funnel, you can actually scale it up. So the important thing when you scale it up that you use bigger tools, because you can't increase the time. So in my experience with A-beta 42, that time is the most important factor for getting nice, pure peptide. So from the time you crush the cells and sonicate them until you have your eluates from the ion exchange, either through filters or onto the ion lyophilizer, it shouldn't be more than like, hopefully 4 hours, but if it's a small batch, it can be 4 hours. If it's a bigger batch, maybe 6 hours, but it should really not take longer time. So to make a bigger batch not take much longer time, then you just increase. So if you use 40 ml resin for a small batch, that gives you 10 mg, and you want 70 mg, you just have to use the bigger size and the bigger, and really make sure that the funnel has a big bottom so that the flow is fast. But otherwise, I mean, it's the same because you're on a macro scale. So there is really no scale up considerations. The thing is, when you come to the size exclusion, you cannot take all in one go. You can never load 70 mg on a column. So you just have to do multiple sizes. So what we do for solid state is that we actually take several FPLCs and then run the first size exclusion on all of them. And it would maybe be 10 rounds. And then we lyophilize all, and then we do the second round for all again in parallel. And when we do fibrils for solid state, we as soon as we have monomers, we put them in a glass flask. So they are starting to form fibrils quiescent. And then as soon as we have the next pile of monomers, we just add them to the same flask. So we keep on adding until we have the 70 mg isolated. Yeah, so that's how we have done it. There's no magic. It's the same. And then we validate. We can either take a little aliquot and validate with thioflavin T fluorescence, or we can take afterwards a little aliquot to validate that there is fibrils. But you also see it when you swirl the flask. So when you make these massive amounts that you need for a solid state NMR, you see them by eye. You see the glittering of the fibrils actually when you swirl the flask.
Great. So let me take another question from Q&A from Patrick Vanderwall. To everyone, to what extent do you see and or use variations in the emission frequency of THT as opposed to just intensity changes? Whoever wants to answer, go ahead.
I could quickly answer no.
Go ahead.
we haven't done this.
Yeah. Oh, you haven't done this. Anybody else?
Same for us here. This is not something that we're typically looking at compared to other dyes or other probes.
Okay.
Same for us.
So sorry, go ahead, Sandrine.
No, same for us. We didn't check, but I don't think so.
Okay, so that's the answer. So nobody has checked it. That's a very good point. Let's take question from Rakesh. Go ahead, ask the question.
Actually, it's not a question, it's a comment. Thank you for great presentation. I think as a disclaimer, for the last 10 years, we don't use thioflavin to follow kinetics. We use it for specific time points. And the reason for it, because when the fibrils form, they are different. Sometimes you have straight fibrils, you have twisted fibrils, sometimes you have clusters of fibrils, and this for sure affect the thioflavin T signal. So my point is, doing the kinetics is not for every lab and everyone. I think the key is to measure, if you are using a small molecule, measure before at specific time points. And one thing was missing from all the curves I saw, except few, is the last end, the dip in thioflavin T. That happens. It dips after a while. You know, you show me 4 hours, 5 hours, but after that it dips. And if you water sonicate it, you recover it. So this is the missing piece. And I'll stop here. Thank you. Great presentations.
So that's a very good point. We do see that in lipid bilayer containing samples. Any heterogeneous system, you do have decrease in the intensity of THT for long times. could be, we thought it could be because of fiber remodeling, or could be THT binding efficiency decreased because of, heterogeneous mixture and so on.
Even annealing of the fiber will affect the thioflavin T, and it's an important step in fiber formation.
Yeah.
And sometimes you see these things that the curves initially rise, so you get like a little hump. And that is probably because it's an indirect method, and you may get actually higher quantum yield for some early fibrils than you get from the later ones, or you may get oligomery structures. So that's why you need a complementary direct methods like monitor by NMR, the monomer consumption, because then this
is not an issue for experts like you and the presenters, but some people who just start in the field, sometimes they get puzzled by it.
Yeah, but it is difficult because what you often want to do with your thioflavin T data is to analyze it. And then if you have this kind of hump, where should you normalize? Should you normalize after the hump or on the hump? I mean, that is a decision you have to take. There is no good solution to that, I think.
So let's take the question from a Q&A folder from Esteban Panazo. Hope I said your name correctly. It's a long question. In case you have a compound with the same emission spectra as THT, and you want to see the behavior in terms of aggregations on synuclein, how we could solve the problem? Because we look many publications with THT and compounds which has the same emission or an absorption spectra, which can be quenched the emission of THT. However, it has been published without the explanation. Is this the competitive binding is talking about, the small molecule and THT?
I think it's mostly talking about overlay in emission and absorption. And basically, like Sara nicely said at the beginning, thioflavin T is an indirect method. So it's a good method sometimes to screen, but I think it's a preliminary data. And in fact, more direct methods are mostly appropriate for that. So it might be a bit longer, but in this case, I think it's just to be careful and to use a more direct method in this way. That's my humble opinion, or using different probes.
Okay, so it does say it's overlay. Yeah. For example, hemoglobins or porphyrins against synuclein aggregating using THT methodology. Okay, let's take a question from Ashutosh Tiwari. Go ahead, Ashutosh.
Yeah, first of all, very nice talks. And I'm also a fellow protein chemist who works on protein aggregation. And it was very interesting to learn with all experiences that you showed. And this is a general comment and a question. Looking at the aggregation kinetics, we all know that protein aggregation is a stochastic process. And it is true that thioflavin T is an indirect method. What I was intrigued, having followed, which all of you must be following, the LLPS literature, liquid-liquid phase separation, that the air-water, air-solvent interface makes a huge difference. And I think that to that point, Steve alluded to the experiment in the cuvette being a little more stable compared to done in the plate. So has anybody tried to do an experiment where, I don't know how it will be done, that you flush with nitrogen or something and then see, because any little perturbation. And if you look at the experiments largely done, even in the LLPS category where people have used shaking, you can find from say 50, 100, 250 to 1000 RPM shaking done, means using different equipments where people are doing, and everybody's getting a kinetics based on that. But the biggest factor, having closely followed what I'm seeing, that this air solvent interface is playing a critical role. So I'm very interested in learning your thoughts about, has anybody looked at that and seen that as a factor in the kinetics experiment and the variation?
Who wants to go first? Steve? Go ahead.
Yeah, I can go. I don't have a really clear answer on that. It's really an important aspect. I think the idea of bubblings might be interesting. I've never thought about that, but yeah, definitely. And basically, I would go back to how sensitive it is, just fibril formation. I mean, even if you're using a pure peptide, as you know, you do the self-assembly in a 1.5 ml Eppendorf tube, and you do the same one in a 5 ml Eppendorf tube, the same surface, and at the end you have different kinetics, and you might have different morphology also. So it's quite sensitive here. Is it just I think it's the surface effect, but I don't have a clear setup in my mind how to address that, but I think it's going to be important. But the idea of bubbling something, I don't know, Sarah, what you think, if you ever tried that.
To have like a line of bub. No, we haven't
exactly.
tried that, but then you would have, then you
Instead
would,
of shaking,
we
but
get a
bubbling,
lot of,
you know, in a controlled manner. So yeah.
So let me clarify the question a little bit. So you can use microcentrifuge tube also to shake sample instead of, say, using 96 well plate. And if the tubes are plated, or the tubes are put horizontally versus vertically, the same volume of protein, people have reported that their aggregation kinetics based on that is very different, and they have attributed to either rotational shaking or whatever. They've used the same identical equipment for the same amount of time and have reported very different aggregation kinetics. And that's why I'm bringing up this question, because in a plate reader, 96 well plate, you have over 200 microliters approximately, and that is quite a bit of surface exposed with very little density compared to, say, a cuvette where you have approximately 700-800 microliter and very little surface exposed on that relatively.
Yeah, we face all this problem for certain systems going from THT plate reader data to NMR tubes. It's a big issue. They don't go exactly the same way. Okay, so maybe related to that, maybe we can address a question related to the well plate transparent versus dark one or black one. Do you guys have any experience? I think Lucie, you have some experience with that. Can you comment on that?
Yes, when I start first, like 15 years ago, we used transparent well plate, but the signal was not it was reproducible, but the signal was less nice. There was some scattering. So then we decided to go to complete black well plate, and then it was better. We decreased the scattering, and it was more, the plateau was more linear, so it was nicer. So we decided to go on with this totally black well plate. And recently I use also a black well plate with a transparent bottom, which finally I think are really nice, but they are much more expensive than the totally black. So then it's really well, at least in Europe, I don't know everywhere, but in Europe it's like maybe the price is 10 times more expensive. But both are nice. Black with transparent transparent bottom and totally black, they are both nice.
There's a comment from Daniel Raleigh for Ashutosh's question, I think, related to that. So I believe David Walk's lab has looked at this, at least in the context of 96 well plates. If not Walk's, then one of his co-workers at Oxford. So, okay, so let me take a question from Q&A. Tom Rothstein asking this question: does sample shaking completely eliminate the measurement interval effect? Does it depend on the nature of sample shaking? Is there an effect of an added Teflon ball on the measurement interval effect? Maybe Sam, you did this, you can answer the question. Sam.
Yeah, I mean, I didn't look like systematically at how the measurement interval affects under non-quiescent conditions, under shaking conditions. I would expect, the agitation in the shaking is generally orbital when you're doing it in the measurement, versus the agitation during the measurement is more linear. And people have shown that there's a difference there. So I don't know if the effect would be as large as it is under the quiescent conditions, but I wouldn't be surprised if there is. something to consider there. I don't know if Dr. Bourgault, if your lab looked at that.
Yeah, that's perfect. Indeed, when you're doing an agitation or either orbital or linear before each reading, it decreased the effect of the number of wells of the interval of measurement. So.
Okay, the next question is from Francis Hita. Thank you all for sharing your experience. I would like to ask, which would be your approach if you are trying to measure a sample that contains another fluorescent molecule apart from THT that may quench the THT signal? Would fluorescence anisotropy that is followed in the plate reader be an option in this case, or there is another approach that is better?
Yeah, I could say something. It sounds like in this case, you cannot use thioflavin T. So either you use a dye with a completely different spectrum, or you can use fluorescence anisotropy. Like for IAPP, you can use tyrosine anisotropy. That was published around year 2000. And that produces nice aggregation curves because the tyrosine gets more restricted in motion in the fibril compared to the monomer. So that's actually a direct method. That's very nice to use. And then you abolish the complication with the other molecule.
Okay, that's good. Let's take another question from Gareth Morgan. Really nice talks. Thank you all. A quick question for the panel and some comments. Any tips for preparing and storing THT solutions? I like black, clear bottom plates because they, I don't know, it's cut off.
We can hear what
Okay,
I do.
Because they are easier to seal.
So what I do like 100 mil or 50 mil of thioflavin T. I dissolve it. So I buy the best one, which I think is Calbiochem. I dissolve it and then I filter it through 200 nanometer filters. First, I wash the filters, of course, to get rid of glycerol, and then I filter it. And then after that, I dilute the stock to measure the concentration because it's too concentrated to measure. And then I aliquote it, like 100 aliquotes, and I put them in my freezer. And then I take out from there.
I dilute it in the water. I also filter it. I dilute it in the water, but then I keep it at 4 degrees in the fridge, not in the freezer.
It's probably okay also in the freezer. It's a stable molecule.
Yeah, but I don't keep more. I keep it like for a year, not more.
Okay, yeah. Yeah, but if it's stable for a year, that is still okay. I put it in. I've never had it in the fridge for it, but it's good to know that you can have it for a whole year.
Yeah, yeah.
So there's a comment from an anonymous attendee. Did anyone put oil on top of the wells? Did anybody? No, right? We surely see that proteins like asin, I don't know what it is, at greater than 200 micromolar in NMR tubes do not aggregate ever.
I think it's alpha-synuclein.
Oh yeah, alpha-synuclein, okay.
Yeah.
Okay, so we have Carmelo on the panel. Go ahead, Carmelo.
I'm here.
Go ahead.
Some question for you about THT assay. The problem with this assay is that the fibril formation is an irreversible process. So if you have, it's an irreversible process, not reversible at equilibrium. And so if you have a small fluctuation of temperature or concentration or other small molecule, can interact, the path change. This is a source of reproducibility. Another source of reproducibility is the nonlinearity response. I will show Sara in a THT measurement. Because when you add, for example, zinc or another proteins, it's possible that the other small molecule quench the fluorescence of THT. I don't know. What do you say about this?
I would just say that it's definitely reversible. Amyloid formation is a self-assembly process. It's not covalent, it's a non-covalent. And things go on and off, and even after you establish equilibrium, you have all processes still going on, but with different rates, of course, and during the process. But there is no such thing as irreversible. Then you have to cross-link it covalently. So if you take fibrils and dilute them to below the critical aggregation concentration, they dissolve. It's just that it takes time, but it's definitely reversible. And for alpha-synuclein, you see this very clearly, because that one is relatively fast exchange even from the fibrils to the monomer state. So if you just dilute alpha-synuclein fibers, you can watch them go away with any technique.
I not agree with you
it's like
because in my experience with the FM measurement, when you observe the fibril, you obtain a distribution of fibril with different shape, length, and so on. You have a distribution, a Gaussian distribution flat, not sharp. So this is typical of irreversible process.
Yeah, but if you don't dilute them, it will change. The whole thing will, yeah.
From my previous experience about folding, when you study blue copper proteins, because blue copper proteins, the unfolding process occur, you have a part at equilibrium and a part not equilibrium where the protein aggregate, form aggregation. And this is irreproducible, this process. Is a delicate process. So if you have a small fluctuation in temperature, because this phenomenon aggregation is an exothermal phenomenon, not endothermic. When you, in fact, when you use agitation, you put in the symbol energy. Is work. And change some condition. This is the origin of irreversibility. Is typical of this process.
It's reversible when you change it. Dilute or change the temperature, they will dissolve. Yeah, but if they are at equilibrium, then they, of course, they constantly form and reform. So if you are at equilibrium, then you can watch them constant, and you have the constant distribution, because whatever dissociates will form again. So you have to change the conditions, either dilute to below the aggregation or just dilute to lower concentration. Some will dissolve. Or you can change the temperature, as you say, because if the solubility is higher at lower temperature, you can just change, lower the temperature, and then part of what you have will dissolve. But I think you also, I didn't hear exactly the question, but part of the question was probably that you can drive the equilibrium. So if you have a self-assembling system and you have a dye, like thioflavin T, that only binds to one side of the equilibrium, of course you will drive the equilibrium over to the other side. The question in each system is you have to find out how much. But that is a physical fact you cannot get around. So of course you drive the equilibrium. I think we're saved a little bit when we do kinetics because the back reaction, as you say, is very slow. But it is an error you have to keep in mind. And that's also why other methods, you need the direct methods as a complementary. Like for alpha-synuclein, it's relatively, I wouldn't say easy, but it has been published by others, not by us, but it's possible to follow with intrinsic fluorescence. So you can just skip thioflavin T altogether.
Sara, I agree with you when you have a symbol system like beta 114 or 42. Yes, when you add another molecule, the system is more complex and not easy straightforward, unfortunately.
Yep, and that's what I said. It's indirect, and you have to keep in mind, and this is an additional complication that you drive the equilibrium. So one also has to think about that, especially if you do solubility studies. If you want to know the equilibrium, if you want to know the exact monomer in equilibrium with fibrils, you have to check that without thioflavin T.
Okay, thanks.
Thank you, Carmelo. Another question is, Steve, you showed temperature-dependent THT binding intensity. Can you give some insights on this? Is it binding affinities increasing, or do your fiber morphologies getting different with the temperature? You showed for one temperature is very high, another one is low. I forgot what peptide, maybe IAPP or A beta, I forgot. Could you comment on that, Steve? Yeah.
Yeah. I mean, we didn't do a thorough investigation on that. We just wanted to look if the how much sensitive the lag phase was changing based on the temperature. So just to address this, but definitely, I mean, if you prepare, if you aggregate at 4 degrees, or if you do an aggregation at 37, you might have different morphology at the end, and of course, you might most likely different THT binding. So the fluorescence might change also. So it's difficult for our setting. I cannot comment based on our experience. That was not the objective here, and I'm trying to. I don't recall a specific paper addressing the specific question. So maybe I don't know if anyone knows the answer here, but I don't have a clear paper in my head looking differently at temperature dependence of THT fluorescence. I don't know anyone.
We all know that, especially when it's very hot in the lab, and if you cannot cool down the experiments, we all know that this increase a lot the aggregation and decrease the lag phase. This is for sure.
Okay.
Because
so we did a temperature dependent study. We didn't specifically look at the tire flame with the intensity as a function of temperature. Well, we looked at the concentration dependent kinetics as a function of temperature. We published that 2018. And then it was quite clear that both primary nucleation and elongation follow typical Arrhenius type behavior. So the rates are higher, the higher the temperature. But secondary nucleation was actually flat. It was even tipping a little bit the other way. So if you go to low temperature and do an aggregation at, for example, 10 degrees instead of 37, you get much sharper curves because secondary nucleation runs as fast as at the other temperatures, but primary nucleation and elongation is slower, so you get much more secondary dominated curves.
I measure the monomer depletion by NMR at different temperature, and it's the kinetics is really different. At 37 degrees, the monomer is I consume all the monomer after like 1 hour or something like this. And when I decrease the temperature to 20 to 10 degrees, then it's much slowly. So, and you have more monomer at the end of the experiment.
Okay, it's 1:30. Thank you all very much. You answered all the questions. Thank you all very much for your time and sharing the data and the experience. It's a wonderful experience and a lot of learning. We'll be in touch. Thanks.
Thank you very much.
Thank you very much. Bye.
Bye.
Thank you.
Okay,
Bye.
thank you very much.
Thanks, Rams.